Beneath the blistering sun and the barren cliffs of the Judean desert, something extraordinary remained hidden for 2,000 years. Silent caves carved into the rugged stone held fragments of a forgotten world, a puzzle waiting to be solved. Few could have imagined that within those shadows lay one of the greatest treasures of human history. This was no ordinary treasure of gold or jewels, but something far more precious: knowledge. Fragile manuscripts written by ancient hands, preserved. Ideas, laws, and stories that shaped civilizations. They waited patiently, untouched by time, until chance brought them back into the light, and it all began, not with archaeologists or scientists, but with a simple shepherd boy and a lost goat. In 1947, a young Bedouin shepherd searching for his goat tossed a stone into a cave near Qumran. Instead of the bleating of his animal, he heard the sound of pottery shattering. Curious, he stepped inside, and what he found would change history forever. Inside lay clay jars filled with ancient scrolls. He couldn't have known it at the time, but these fragile manuscripts were the oldest surviving copies of the Hebrew Bible, along with a wealth of other texts never seen before. The boy's simple act had opened a door into the distant past. Soon word spread of his discovery, and archaeologists rushed to the desert. What began as a shepherd's accident became the start of one of the most important archaeological excavations of the 20th century. Over the following years, 11 caves near Qumran were uncovered, each holding pieces of an ancient library. More than 1,000 manuscripts were recovered, ranging from biblical texts to religious rules, from prayers to philosophical writings. Together, they became known as the Dead Sea Scrolls. For scholars, these scrolls were priceless. Written between the 3rd century BC and the 1st century AD, they offered a direct glimpse into a pivotal time, when Judaism was evolving and Christianity was about to emerge. No other discovery had brought us so close to the thoughts and beliefs of that era, but the scrolls came with a challenge. They were not found intact. Time had reduced them to over 25,000 fragments, many no larger than a coin. To unlock their secrets, researchers faced the daunting task of piecing them back together, like an enormous jigsaw puzzle missing half its pieces. Imagine standing before a table covered with 25,000 scraps of parchment, none labeled, many faded beyond recognition. That was the challenge awaiting the first scholars of the Dead Sea Scrolls. Each fragment had to be carefully examined, matched by handwriting, language, and content. For decades, experts argued over the origins of these scrolls. Were they the work of a single isolated group in Qumran, or were they a collection of texts gathered from many different Jewish communities across Judea? The fragments refused to give up their secrets easily. It seemed as though the mystery of their true origin might never be solved. That is, until science introduced a tool that the ancient scribes themselves could never have imagined: DNA analysis. The identity of the scroll's authors has long been one of history's greatest debates. Some scholars believed they were written exclusively by the Essenes, a strict Jewish sect living at Qumran. Others argued they came from multiple groups: Pharisees, Sadducees, and even unknown scribes scattered across Judea. Each theory had evidence but also contradictions. The content of the scrolls suggested diverse traditions, yet their location in Qumran pointed to a single community. Without solid proof, the debate raged on for decades. But then, scientists realized that the answer might not lie in the ink or the handwriting, but in the material itself. The parchment was made of animal skin, and inside those skins, even after 2,000 years, fragments of DNA still remained. At first glance, the scrolls looked like simple parchments, fragile but ordinary. Yet to molecular biologists, they were something else entirely: a hidden archive of biological information. Each piece of parchment was made from animal skin, and every skin carried the ghost of its owner, DNA preserved for 2,000 years. This was revolutionary. For the first time, scientists could analyze not just what was written, but the very material on which it was written. By studying these genetic fingerprints, researchers could begin to trace where the scrolls came from, who might have produced them, and how far they traveled. The idea was simple but daring. Let the animals themselves tell the story of the scrolls. But there was a problem. Ancient DNA is delicate, fragmented, and easily contaminated by modern touch. Generations of scholars and conservators had handled the scrolls, leaving behind traces of their own DNA. Extracting authentic ancient sequences would require near-perfect precision. To solve this, scientists in Sweden worked inside ultra-clean laboratories, using airlocks, UV lights, and specialized tools. They collected microscopic dust from blank areas of the scrolls, ensuring the manuscripts themselves remained untouched. Through a process called silica-based extraction, they were able to capture ancient DNA fragments like magnets pulling metal. Piece by piece, the genetic code of 2,000-year-old sheep, goats, and even cattle began to reappear. Most of the scrolls were written on sheepskin, perfectly suited to the dry Judean desert. But then came a surprise. 2 fragments, both from the book of Jeremiah, were made of cowhide. At first it seemed like a minor detail, but in archaeology, details can change everything. Cows could not have been raised in Qumran. The desert was too harsh, too dry, with no pastures to sustain them. There was also no evidence of cowhides being processed there. This meant one thing: these cowhide scrolls had to come from elsewhere. The discovery shattered the long-standing belief that the Qumran community wrote all the scrolls themselves. Instead, it suggested something far more intriguing. Qumran was not an isolated sect, but a center where texts from across Judea were gathered, studied, and preserved. If Qumran was not a fortress of isolation, then what was it? The DNA evidence pointed to a new possibility. Qumran was more like a library, a hub of intellectual exchange where manuscripts from different regions came together. This changes the entire narrative. Instead of a single group writing in secrecy, we now see a picture of collaboration and diversity. Scrolls from fertile regions like the Jordan Valley likely traveled to Qumran, where they were stored alongside locally written texts. In this new light, Qumran becomes less a hidden monastery and more a crossroads of Jewish thought, preserving voices from across Judea during one of the most turbulent and transformative periods in history. Among the many manuscripts, one stood out: the Songs of the Sabbath Sacrifice. Copies were found in Qumran, but another copy appeared miles away at Masada, the fortress famous for its dramatic last stand against Rome. This sparked a debate. Was the text exclusive to the Qumran sect, carried to Masada by wandering scribes? Or was it a work so influential that it circulated widely among different Jewish groups? DNA held the answer. The Qumran copies were written on skins from genetically related sheep, all from the same flock. The Masada copy, however, came from a completely different lineage. This meant it wasn't imported from Qumran. It was independently created there. The Songs of the Sabbath Sacrifice were not the property of one sect, but a shared spiritual tradition treasured across Judea. Not every scroll fragment truly belonged to Qumran. Some pieces, acquired through antiquities dealers rather than excavations, raised suspicions. One fragment in particular, a simple legal document acknowledging a debt, seemed completely out of place among the religious texts. Genetic analysis confirmed the doubts. The parchment's DNA did not match the genetic profile of Qumran's local animals. It was an outsider, a fragment mistakenly mixed into the collection. Another piece from the book of Isaiah also proved to be a genetic outlier, suggesting yet another origin far from Qumran. For the first time, science could separate authentic Qumran manuscripts from misplaced ones, bringing clarity to decades of confusion and reshaping the very catalogue of the Dead Sea Scrolls. The DNA evidence transformed everything we thought we knew. The Dead Sea Scrolls were not the work of a single, isolated sect hidden away in the desert. They were the product of many communities, reflecting the diversity and complexity of Jewish life during the Hellenistic and Roman periods. Some texts were written in Qumran itself, shaped by its unique traditions. Others were carried in from distant valleys, copied by different scribes, and preserved together in this desert library. Each fragment is both a physical artifact and a genetic witness, holding the memory of the animals that gave their skins, the scribes who wrote on them, and the communities that treasured their words. And yet the story is far from finished. Thousands of fragments remain, each carrying secrets still locked within. With every new discovery, science continues to peel back the layers of time, bringing us closer to understanding not only the Dead Sea Scrolls, but the world that gave birth to them.